think it's like bioengineering.
So let's go ahead and get started.
Yeah.
Okay, so last day of the workshop, so we're going to start off with Jen coming right here from UC Berkeley.
I'd like to thank the organizers for inviting me to present here today. So I keep using this title, but changing the talk a bit so I don't have to think too hard. Is this working still? Lagged. Oh, wow. Super laggy. I'm not going to do that. So I think, we've already heard a lot about proteins. And so some of the stuff I'm going to mention, you've heard in bits and pieces, and I'll put it sort of in my own viewpoint. And I don't think I need to motivate to this crowd why protein, and so it's right, this is gonna be a whole talk about protein engineering and some peripheral items as well. But of course, we use this all over the place for antibody therapeutics, for CRISPR gene editing, for biosequestration. A lot of this is already in play now, and a lot of this is also aspirational. And this is, I think, an extremely exciting time for protein engineering. And I think I also don't need to tell this crowd that if you just abstract away everything and look at this as a computer scientist perspective, where you're trying to design, at the end of the day, if you're doing protein engineering, you need to specify a sequence. Maybe you do it through a 3D structure or something, but you need to tell the person in the lab what the sequence is. It's a 20-letter alphabet usually. And so even if you have a fairly short protein that's just of length 50, which would be a tiny protein, you're already near, in terms of the number of combinations just with that alphabet in raw size, near the number of atoms in the universe. And so fundamentally, this is just some massive combinatorial space. But of course, nature has actually evolved proteins and through this crazy space. And so we know there's a proof of principle that this can be done. That's why we're all sitting here listening and alive and breathing. But of course, this took millions of years. And so we don't want to wait millions of years to get the proteins that we want. And therefore, over the past few decades, a number of protein engineering strategies have emerged. And I think probably everybody here has heard of kind of the two main ones that have been driving the field that are quite complementary and different and good for different things. So there's this sort of Rosetta-based energy function computation-based approaches that Baker's lab has spearheaded for many years. It says almost rest in peace. So you don't hear people publishing about these energy functions anymore, but actually I know people who are using them in industry or in academia. The places I know they've been used recently are for example, antibodies. But we now know from when Mohammed gave his talk and from the AlphaFold 3 that maybe it is finally rest in peace, but let's see. And then of course from the completely other side, the wet lab side, which is sometimes used a little bit of computation to help itself, but is sort of primarily iterative wet lab cycle and got the Nobel Prize in 2018, is directed evolution. And of course, the reason we're all talking about this now is that machine learning has started really in a fairly fundamental way to integrate with all of these. And I think these are all kind of becoming one and the same thing is because at the end of the day, most people still do directed evolution, even if they're using a fancy diffusion model to generate, then they need to still get it to where they want it to be. Anyway, so I think all these pieces are kind of coming together, even though certain people take a kind of purist viewpoint on wanting to do it one way. And so a question I often get, even before AlphaFold 3 came out, is, well, isn't your problem solved? Don't we know how to do protein engineering? And of course, AlphaFold 2 and 3 is an extremely useful tool, but it doesn't sort of fundamentally solve the general protein engineering problem. And the reason for that is that it's a relationship between structure and sequence, right? You give it a sequence, it tells you the structure. People have inverted it into these so-called inverse folding models where you could specify the structure and get a sequence or a distribution of sequences. But in either case, what's actually missing is that the functionality of the protein we don't know. If it's a purely binding mediated function as opposed to say catalysis, then maybe we can get quite far with this if we happen to know what structure binds. But in general, we don't actually know what we want, either defined by the structure or the sequence. And so we need somehow to bring function into the mix. And so no matter how rich and sophisticated this machinery comes along in terms of diffusion models on backbones and inverse folding, somehow we need to get the functionality in there. And I think that's kind of, I don't think it's totally overlooked, but people like to sweep it under the rug a little bit. So I started working in this field maybe roughly 6 years ago, and I was very disoriented. I would like read this paper, and it would say it solved protein engineering, and then I'd read one that was doing something different and said it solved protein engineering, and I was like, what is protein? What are these people doing? And so the way I think about things now, which I kind of got a little bit from my former student, Chloe, is in terms of, at least as a machine learning person, that there's a set of prediction tasks, and on the next slide, a set of design tasks that as a kind of machine learning person, I might like to categorize things in. And so I would say you could, this is just a choice of how to view it, but like you might say, if you give me a sequence, you might want to predict the function, or you might want to predict the structure. And these would be sort of, if you're doing it with machine learning, a machine learning predictive model, for example. And then on the design end, you might say, well, you tell me the structure, I'm going to give you the structure, and you design the sequence. I mean, you could kind of call it prediction, but when people talk about design, it's usually one of these tasks. It says, I tell you the function value I want, and you tell me maybe the backbone coordinates, and then from there, you tell me the sequence. Or maybe I tell you the backbone coordinates and you design the sequence. But these are the sort of the class of problems that people usually mean when they talk about protein engineering or designing a protein. And they may get combined in sequence. Like as we've seen, there's often this way of first getting a structure and then after the fact, getting a sequence conditioned on that structure. And I should say, as with all these talks, please feel free to interrupt. I don't expect to get through all my slides. Okay, so what are some of the trends that have been going on for like the last 5 years? They're in a lot of ways, they're quite similar to the broader part of the broader field of machine learning, but there's definitely very special things related to biology and the sciences owing to lack of data. It's not all on the web and things like this, but roughly speaking, we have, these people are chasing representation learning. So getting some big unsupervised data set, trying to see, like, can I understand, say, something about protein sequences in terms of a latent space, in the hope that that latent space captures correlations that will be helpful for me, for example, to use as a featureization on some limited number of supervised data, in the hope that maybe the representation actually captures epistasis that I could not have found just from my limited supervised data. And so no matter, I think no matter whether it's a transformer, it's mass language model, whatever, like at the end of the day, this is really, at the very least, you're approximating a density and you're doing density estimation. Okay, and then another one is generative models for sequences. Or density models for sequences, but and often we'd like to have conditional generative models for sequences. So for one way of conditioning would be these so-called inverse folding models, where you condition on a backbone structure and then ask for a distribution of sequences that are likely to fold into that structure. There's this idea that you'd like to have a natural language interface and just tell it with some kind of control tag what the sequence is. Maybe in terms of protein family, we can probably do that now, but not in some general protein engineering way. Okay, and then of course we have conditional generative models for structure, which John talked a lot about with his beautiful chroma work. I've stolen a picture here from him. Well, it's cited, so I didn't really steal it. And I think like this is beautiful work that is changing the world, and we know there's also the RF diffusion kind of version. But at the end of the day, like if you want to do guided diffusion, to get the condition, you want to condition on some target property of your protein. If it's an alphabet, then you can say, I want it to look like an A. You can design the structure to look like, and you can do that. But if you don't know enough about the function, then you cannot provide the sort of guidance in the diffusion space. And so I'm not in any way trying to disparage, like these are fantastic, and I think these are changing the world. But again, we need to somehow understand the relationship between the function we desire, such as catalytic activity, and say the backbone, for example. And just a little plug, if you're a student want to understand a bit about generative models in this space, we have a little primer out quite recently. So another one is how do we actually just estimate these functions that go from sequence to function? This is sort of the missing, one version of the missing ingredient. And often we collaborate with quite a number of labs. And in the beginning of our engineering campaigns, we don't usually have any labeled data at all. So we need to start with some sort of zero-shot model to design the first library. And one of the key ingredients for proteins is making use of evolutionary information. So we're not typically working with purely synthetic proteins, we're usually starting in some part of known natural protein space, and then we can kind of ask what are related proteins, and then there's a whole field of work of how do I squeeze out sort of relevant evolutionary information from these data. And you can do that in a family-specific way where you kind of search through a database for like evolutionary related proteins and, for example, build a density model on those and hope that if you're sort of in distribution for that density model, that to the extent you are is the extent to which you'll exhibit the same properties as the sort of seed sequence you use to curate that set. And then we also know these sort of pan-protein models like ESM, where you train it on all proteins families possible. And I think a reasonable way to think of those models is they're a giant mixture of family-specific models that get to share some information and happen to be convenient because you just train it on all proteins and you're kind of done. But I think we know there are still spaces where protein-specific models actually outperform these sort of giant pan-protein models. And I think there are still interesting questions to ask there. And so, right, and then, so I think I updated this. And another thing people have been trying to do, and Mohammed, who I think he's not here today, has been driving some of this, is trying to fill in some of the holes. And I guess in some sense, a lot of the molecular dynamics people that have been here, which is not an area I know too much about, but enjoyed listening to the talks. But we at the end of the day, AlphaFold 2 and 3 is like, it's just essentially predicting static structures, not dynamics. And we know that dynamics are super important. And so this is sort of the wide open, important area of protein structure prediction, if you will. As Mohammed pointed out, that especially with AlphaFold 3, like we're getting pretty good at protein-protein interaction and multi-protein. And there's some advances in binding to DNA, RNA, but I think there's still a lot of room to be made there. And then one of the key things, which, and I can't wait to hear next time Mohammed talks, because I know he's charging hard on this idea of, can I get models to work where this like I give it a sequence that doesn't actually have many evolutionary neighbors in the natural world. So we know that those kinds of proteins don't work well in AlphaFold. That's because at the end of the day, AlphaFold's not fundamentally doing physics. It's actually really depending quite heavily on evolutionary information. Okay, so that's sort of my kind of how I've been learning to think about this space as I started to walk into it a few years ago. So now I'm going to give you an overview of like some of the flavor of work that my group started with and kind of the directions we got pulled into, and then the next two things I'll go into a bit more detail on two projects. This is going to be like flyby kind of ideas, give you a sense, and you can go find the papers. And then this is actually a new preprint that Bear and Stefan and Hunter have just put out, and they're all here. So when I confuse the hell out of you, you can go talk to them at the break. And then if there's time, I'll tell you a little bit about a kind of what looks like a simple engineering project with kind of simple machine learning, but I think we're one of the few people that just did it really cleanly and thoughtfully, and I think we did something really useful, and I don't think there's a lot of that, so I want to tell you about that project. Okay, so this is kind of the picture that quickly emerged when I started working with my first student, David Brooks, who's also been around. I don't think he's here today. I think Sam is here. His boss probably didn't let him come. Okay. And so like David and I started working on this idea of like, if I have a predictive machine learning model that goes from say sequence to property, and now I want to find the sequence that maximizes the property well beyond, as far as I can, outside of what I've seen in the training data set, like how do I do that? And so at the end of the day, we thought this was kind of a combinatorial optimization problem. You give me some like predictor function that goes from, oh yeah, sorry, this is not yet proteins, that goes from sequence to property, and then kind of invert it in a way that I get the sequence that gives me the desired highest property. And what we quickly learned is that there's major pathologies when you do this, which a number of people here have spoken about in different ways. And so this is how I think about it. I think about it as the banana problem. And so in computer vision, this had already been done when we were thinking about the spaces. Someone trains a state-of-the-art neural network for, at the time, this is now pretty old, and I apologize that I somehow lost the reference along the way, but you have this trained neural network where you give it an image and it tells you how, like probability banana, kiwi, zebra, whatever, hundreds of classes, right? And what the computer vision people want to do is to say, well, let me probe at this neural network and ask, what does it think a banana is once I've trained it and it has these state-of-the-art results? And so what they do actually mechanistically looks like one version of protein engineering, the way we started to think about it, which is take this trained network that could be, for example, from sequence to some property of the protein, and take the trained network, fix a node. In our case, we might just have one node catalytic efficiency, and it might be regression. In their case, it's a classifier. So set the banana node to 1, set the other nodes to 0, and essentially backpropagate, starting with a random image, and then find the most banana-like image this neural network believes. And of course, it's a non-convex problem. You don't know where you get, but you get somewhere reasonable. And this is kind of the most reasonable thing you can extract. It's probably not a global optima, but what you see is that you get something very, so this is like the banana design problem, if you will, in analogy to the protein design problem, where I might have said, give me high catalytic efficiency, and you tell me the sequence, right? And so, but when someone shows me a sequence, I'm not trained as a biologist or a protein person, so I can't tell much from it. But so that's maybe why I like the banana, is like, it's very clear that it's done something very plausible. It has the right colors, right kind of contours, and yet it's like complete nonsense, right? I can't go 3D print this and put it on the table. So now if I'm doing this with proteins and this kind of thing happens, like obviously I'm in trouble. So what's going on? It's this sort of well-known fact that these, especially these very high capacity models, do well near the training data. They have massive blind spots that can be attacked adversarially. And when we do design, we're sort of accidentally being adversarial by squeezing it into parts of the space because we want to find proteins with properties we've never seen before. If you're trying to do what I would call extrapolative design, if you're trying to stay within your training data, you may very well be okay. But usually in protein engineering, we want to achieve some protein property we've never seen before. And right, so correspondingly, it's very likely that, like when David started running this and he said, I'm no protein scientist, but I bet you this protein's not going to fold. Like, this just looks weird to me. And so this was the start of our thinking about this. And we have a number of papers. And the first one, I'm not going to go into detail, but like basically, what's the kind of core issue here? And it's like an unsolvable problem, if you will. And so the question is, how do you navigate this unsolvable problem? All the real interesting problems are unsolvable. And the problem is really that in this sort of extrapolative design, I'm trying to navigate a tension between pushing my model beyond what it's seen into some notion of extrapolative space. And at the same time, I know that these models can't extrapolate very well. I don't quite know how far they can extrapolate, but I want to squeeze them for all they're worth and somehow control my risk as I do it. And so you can put on sort of different formalized hats to try to control this risk in a rational manner. I don't think there's like one perfect way to do this, but so we've been thinking of different ways. And so some of the themes that emerge are, well, the very first one was simply, actually it's quite funny because there were, I don't think people are doing this now, but at the time we put out our first paper at ICML on this topic, which was basically saying, if you have a trained neural network and you invert it, like I did with the banana problem, you just treat it as a combinatorial optimization problem on the learned function, like this is silly. Like you're not going to get somewhere because you're squeezing it too far. And so basically what this paper is about is a technical framework with which to bring in some sort of soft trust region, which, you know, this is not a miracle cure. How do you define that? One way to define a sort of soft trust region you might want to pay attention to would be the density of the training data you used to learn that function. Or it might be the density of something like ESM that says this is like you lie in the kind of natural protein space that probably folds, for example. But then, anyway, so we've been thinking in this space in quite a few different ways. And so Hunter has this beautiful paper. It got an Editor's Choice Award. Go Hunter. And it's this sort of like it's this very nice intuition. And basically, he developed the technical machinery to instantiate it. And in many of these cases, like there are difficulties to actually practically instantiate this machinery, but just developing the machinery and thinking in this way already really helps you to just think clearly about your problem and how you might want to instantiate it to get this machinery to work. So what is this paper about? This paper says basically that we know that historically for a lot of biophysical properties, we had models that were not data-driven. They were more biophysics approximation-driven. And because they're biophysics-based approximation-driven, they don't sort of have some like area near a training data where they're particularly good and then sort of fall off quite rapidly, right? Instead, they're kind of probably uniformly good over much of the space. However, if you go and for that same biophysical property, you get some amount of training data and you now train a machine learning model, then near the training data, this model is probably going to beat the hell out of that biophysics-based model because if we're kind of reasonably careful with what we do. And now there's this sort of what happens is you walk away from the training data. At some point, you want to probably kick into the biophysical model because you trust it more because it's so far from the training data. And as you get closer to the training data, and so you want to kind of coherently blend these models such that it does the right thing at the right time relative to your sort of distance to the training data, if you will. And so fundamentally, if you want to solve this problem well, you need to be able to estimate your epistemic uncertainty, which is the uncertainty you get owing to just lack of having had data to estimate your model. And so Hunter basically went through a whole bunch of math and machinery and got out these very nice, intuitive updates at the end that basically say, if you train your machine learning model with some sort of approximate Bayesian neural network, whatever works for you, and you have some sort of function valued prior in a pointwise manner on the space arising from a biophysical model, then you can just combine these sort of post hoc in this nice way that will gracefully trade off between the two, blend them together in the right way in different parts of the space. So, and you can look in there for more details. And then I'll just mention on a third area, so this is driven by Clara Fan-Jang, who's actually going to be speaking this afternoon, I think much more broadly on the overall topic, not necessarily this paper. She's she's a wonderful speaker, so I hope you stay. And so she, in the pandemic, got interested in who's sort of conformal prediction. Good crowd. I think this is a beautiful paper. I also think and Clara is the one who taught me the weaknesses of conformal prediction. So there's a lot of claims that conformal prediction is distribution free. It's doesn't require asymptotics, but there's some other stuff that gets swept under the rug. But fundamentally, conformal prediction is a way to make uncertainty estimates on predictions. So what you would really like is to have a predictive model, and I give you this sequence, and you tell me the binding affinity with some sort of confidence interval you can trust. And so often when people talk about conformal prediction, they present it as though it gives you exact finite sample, non-asymptotic guarantees for that. And it does, but doesn't quite. What it actually gives you guarantees for is the expectation over all training and all test points. It can't give it to you for just this one point. And so one should be careful in using it for that reason. But Clara found areas where with the expectation, you could still ask interesting questions. And the expectation would come up in doing library design where you have many, many test points. So you're trying to design a library of thousands of proteins potentially. And so this expectation, at least over the test points, is sort of actually quite sensible in this case. But I'll leave this topic to her. But actually, she and I are fans of writing. We love writing together. And we got invited to write. I don't know what they asked us to write about, but we just put all the things we'd love to think and talk about that we didn't know how to put into a paper, which is basically all of these topics of how do you think about the tension between extrapolation and squeezing your model and trusting the model. And so it's sort of a general conversation or discussion, but it is anchored in protein engineering. However, I think a lot of the ideas there apply more generally to machine learning based design. Okay. Wow, this is a good amount of time. Okay, so I'm going to tell you a few more of these sort of vignettes. Just if it's interesting, then you can go follow up with me or the papers or whoever I tell you is doing it. Actually, I saw Amirali had registered, so maybe he's attending remotely, or maybe he'll watch later. But Amirali kind of pulled us into this area. He comes from an information theory background in compressed sensing, and he was a postdoc not in my group, and he came up to me and he said, "Oh, I'm doing this kind of thing where I go back and forth between using a Walsh-Hadamard basis projection and using a neural network and he had this kind of crazy algorithm." And I said, "I think that maybe what you're doing is you're regularizing the neural network in the space of the Walsh-Hadamard basis." And if you haven't heard of the Walsh-Hadamard basis, it's basically like a Fourier basis, but it's for discrete data. And so instead of getting higher and higher frequencies, you get higher and higher order interactions, which is like a protein scientist you could think of as like higher order epistasis. And so in compressed sensing, they have some very efficient algorithms. And I said, you should talk to my very smart student, Hunter. And Hunter actually figured out kind of what Amirali was doing and made a clean algorithm that is truly actually doing this regularization of neural, and if you think, I think this is actually quite an interesting problem. So we're still working in this general space beyond this, that we haven't yet published anything more than Amirali's paper. But there's this idea that when you use a neural network, you can't regularize in some intuitive space that you know about. And maybe you don't often know about an intuitive space, but although I guess in images you have this idea of the frequency spectrum, and maybe there's some work there, but in protein space you have this notion of the epistatic landscape and how high order the terms are. And do you believe that a fifth order epistatic term is as likely as a first order epistatic term? And like, no, you don't. And we also don't have the gobs and gobs of data that people have in other fields. So we better actually, I think, well, there's this, I guess, that plays into should we put in inductive biases or not? I think when we're in very small data regimes, especially, that we should probably put in inductive biases, but I am also intrigued about how we should turn them on to control the dynamics of the optimization and not screw ourselves up. But anyway, so that's basically what this paper is about. It's saying, how can I regularize a neural network that goes from, say, sequence to scalar in a space that I can think about as the epistatic landscape and impose sort of general beliefs about that space, kind of like as general almost as like an L1 loss, but informed by notions of epistasis and what we believe to be in these functions. The second one here is an accidental paper. Maybe I won't talk about it in the interest of time. And the third one, this is like one of my favorite papers. It took forever to get out, but and it's a sort of simple but powerful story about protein engineering. So it's using kind of a lot of the ideas that I've talked about or thought about, and it's like a 3 or 4 year collaboration led by David Brooks and then Akosua Buzia, who have both since graduated and now work for Sam at Dino. And so I'm going to talk about that. And what I would say is also in general, my group likes to kind of think a bit pie in the sky methodologically, hoping we're doing something that can truly be useful, but also to just help us think about the space and what tools are out there. And so, but then we also like to keep ourselves honest and have the opportunity to really test stuff in the labs. I don't have a wet lab, so we partner with quite a few wet labs, and some of these collaborations have been going on for many years. Some are a bit more recent. And so the most established one is the one with David Schaefer that I'd like to talk about. But just so that I don't run out of time, I'm gonna do things in a weird order because I wanna talk about the work of Bear and Stefan and Hunter. So I'm gonna come back to this, and therefore I'm dragging you a little bit all over the place. Okay, so now I'm going to talk about is what I call, this is a new, by the way, I was still fiddling with the slides as it was getting plugged in, and so it's a little bit on the rough end, or it could be, I don't remember what's in it, we'll see. So conditioning for design. So what does conditioning for design mean? So I think we've sort of heard this theme all over the place. So it's often the case that we have some unconditional generative model, say over sequences or over structures. And then, but usually this is not the thing that helps us in science, right? At the end of the day, an unconditional generative model is basically never the end goal modeling wise. It's, if anything, it's a conditional generative model. And so I have, for example, imagine this is the setup, is I have an unconditional model and I would like to condition it on some property y. Could be a class, or it could be some sort of event defined by a regression model. And imagine that I also have access to some supervised data for the sort of conditioning variable y, or that I've already, me or someone else has built a model on those data, or it's a biophysics-based model. And so, so how can I kind of put these two pieces together, an unconditional model and some data or a supervised model, to get this conditional model? And this comes up all over the place, so I've sort of instantiated this with one example. I said, suppose these were protein sequences, and I wanted to condition on structure. But in fact, we see this all over the place. And so like another, just for example, thing that we've thought a little bit about is if this was a small molecule library, like a combinatorial synthesis library, like enamine, and we wanted to condition on some desired chemical property, given some ability to estimate the, like for example, like imagine it was the affinity binding affinity. Like, how could I get this conditional model out? And so I'd say fundamentally, there's 3 ways that you can do this. The first is actually just say, I don't care that I had an unconditional model and these data, like, let me just take all the data I have. And to do this, you'll also need the supervised labels in there. And you're just gonna say, I'm gonna just start from scratch and just bake right in the conditioning variables into my generative model. And you can totally do that. Sometimes people like to blend that with the unconditional model to regularize it. One downside is, like, now you're locked into those conditioning variables, but there can also be some positive sides. Okay, another strategy is to start with some, you've spent a lot of work on getting some unconditional model, perhaps over protein sequences, and now you say, okay, now I'm going to update that model by making calls to a predictive model for the property I care about that I want to condition on. And so, in fact, when we wrote this first paper that I mentioned, which is actually, the title is called Conditioning for Adaptive Sampling, which was actually the thing that I mentioned had a soft trust region. Turns out you can think about this paper in quite a few different ways. And in fact, one way you can think about it is actually quite analogous to what DPO is doing. So this direct preference optimization is starting with an unconditional model and then trying, in the conventional case, to like align it with human, goodness or whatever. But this idea is getting repurposed to do conditioning in different ways. And that conditioning is based on comparing two outcomes and saying which one is better, and trying to make sure that you can kind of pull those apart. And it's not actually paying attention to like the units. And if you were to apply DPO to say something with binding affinity prediction, like, it wouldn't actually understand the true difference of the units of the binding affinity. It would kind of throw those out. And I think that Grant has worked in this space. I haven't read his paper and I didn't completely understand the talk, so he should also be cited here. But I was like, I don't even yet know exactly what you did. I see. Okay. So, and I would say similarly, like, our algorithm here also fixes this problem, because it's actually just using the real values there. But what I would also say is that was fundamentally, in either case here, you are having to update that unconditional model, like, and that's fine, like, and that can be totally fine and powerful. But I think John's the one that really tweaked me to this idea here, and he's not the first to observe it, but I loved his paper, and he spoke about it quite eloquently and clearly, which is this idea that in some models, and for example, the diffusion models, you can spend all this money and resources building a fantastic unconditional model, and then you can keep repurposing that model without ever changing it, to mix and match with different types of desired conditioning, by a plug and play kind of mechanism that diffusion enables. And so I think this one is quite special for that reason. I also think there can be downsides to this plug and play that haven't been explored, and I haven't personally. But so I'm in no way saying that's the thing we should do, but it definitely has that nice capability. So, okay, so this is contentious, and I'll admit I didn't fully think it through as like kind of typed quickly this morning. But I think that unless you're baking in your conditioning, meaning you're directly putting the conditioning variable into the generative model, I think fundamentally you are, even if you don't know it, you're trying to use Bayes' rule. And you maybe are using approximation to it. And I'm happy to get pushback on this, because this is not a rigorously derived statement, but I think it's probably likely to be true. And then I'm not very hip or aware of stuff, so I stole this from Hunter, who had it in his short talk the other day, which is that when you want to use Bayes' rule, I don't even understand it, but if you guys are laughing, I'm very happy. I should have Googled it, right? So, right, but this is always, like, in the old days, we built conjugate models and we get the normalization constant free. Like, who's heard of that in the last 5 years, right? Like, no, I don't know, nobody, basically, right? So this thing is always a pain in the neck. It's a pain, so we don't want it. And so this is sort of the beauty of classifier-guided diffusion. So if you're not, even though we've had a lot of talk about diffusion models, if you haven't heard much about these, I'm going to give you like a 1 minute intro. So sort of, I think you can think of diffusion and score-based models as the same thing. And so they're doing density modeling. They're just trying to learn P of X, but they're doing it in this weird way because it turns out to be powerful, is they're, instead of learning the density, they're learning the gradient of the density with some parameterized neural network. Fundamentally, that's what they're doing. And so now instead of sampling directly or through some MCMC, you're using some sort of like pseudo diffusion process or some iterative process following like a probability flow or something. But at the end of the day, these models are using the gradient of, or the score, this is the score called the score function. And so to sample, you start in space and then you follow kind of more or less this gradient in some noisy way, and then you get your sample. But so like, it's a pain in a lot of ways, but one of the true, it's powerful, but one of the really fantastic, again, this is, I'm even citing John here, this is actually cut and paste from his SI. Do you recognize it? Yeah, I mean, anybody could have written it, but since I was lazy and I cut and pasted, I had to cite you. So, so what happens is when you push the gradient operator through Bayes' rule, you end up with the gradient of the normalizing constant. And the key here is it's the gradient with respect to the space that you're kind of doing generation in. So for us, it might be like if it's a general model over sequences and we want a conditional model over sequences, this is with respect to the sequences. Of course, that's discrete, so that's tricky. Let's think of just images for now. And because the normalizing constant doesn't depend on the images, this just falls out. And so this is sort of why we get this elegant plug and play nature of being able to take an unconditional diffusion model and then just kind of, what happens is instead of just following this gradient as you would in the unconditional case, you just literally add this beautiful guidance gradient. And it's a little tricky because you have to train this classifier to understand the dynamics of the diffusion and so forth. But fundamentally, you get to totally reuse the like unconditional model. And so that's quite beautiful. So that's nice. However, all this diffusion stuff started in real valued space with like images which get treated as real valued. And so, but in the sciences, we often have discrete spaces we'd like to do these kinds of things for. So just as some examples, either sequences or graphs, and in fact, text as well, though we don't think much about text. And so if you have discrete sequences, then the gradient of the density with respect to these discrete objects is not going to help you move around your probability space. And so this has posed quite a challenge for people trying to see, can we leverage the sort of magic and wonderful outcomes of these diffusion and flow-based models to work in the space. And sort of, if you just naively try to reproduce, just use that machinery, you can't do it because that gradient doesn't really work. And more importantly for this talk is that you cannot get any guidance for the same reason. And so that's what we're going to talk about. So people haven't really tackled the guidance issue. They have said, well, how can we get diffusion and flow-based models on discrete state spaces like sequences, for example? And so one thing you can do is you can just kind of relax into a continuous space and then use the same machinery, snap back, and hope that that's useful. Stark et al. Have something where they said, well, we want to be in discrete space. We're going to diffuse on the multinomial space of the distribution, which is like real valued but has some constraints. And then once we diffuse the probability parameters, then we can sort of sample from it. But I think, arguably, and I think it's my students who've convinced me. You can tell me if I took the wrong message away. But I think one of the kind of nicest and perhaps most useful and general frameworks has been led by Campbell et al. In two different papers, one based on diffusion, one based on flow matching, and these use question, yeah.
High level question.
Yeah.
So, like, what is the perceived advantage of having a diffusion-like model on a finite state?
Oh, I totally agree. So
autoregressive one.
Yeah. I think this is exactly the right question. In fact, for months and months, I kept saying to my students, I'm skeptical of these models. Why would we use this on a discrete space? Because like my intuition is that for a real valued space, when you use these approaches, you're kind of touching everything at once, a little amount, and moving. And you cannot touch discrete objects a little amount, and you maybe don't want to touch all of them either. So I think this is a great question that, and I always push back, and then Hunter always gives me a very nice answer. And so I think what you could say, and I'm just parroting Hunter here, so he can just tell me if I've got it wrong, but is that like you can do regressive or order agnostic autoregressive, and so you might think like, well, those are good enough. And I guess the issue is that you want to pick a good order. And so even if it's order agnostic, I think work could be done to put in there to pick the order in a sensible way. But I think what these models get you, and again, this is just like I'm taking this from my students who taught me a lot of this, is that fundamentally, if you do this well, it picks the right order in a sense. But I personally think that they're fundamentally different, and the wins we may get are coming from different reasons, and they're quite different objects. And I personally think it's a bit misleading to say these are diffusion models, because I think they're really different beasts. Is that?
Yeah, I mean, I think there are definitely some algorithmic things that I can imagine you could do quite differently with a diffusion-like framework for, as opposed to a transformer. But it's hard for me to imagine that, one would clearly outperform the other.
I think one other advantage that some of these models have is a common issue with autoregressive generation, particularly for conditional generation, is it commits too early and can't correct these mistakes. And these diffusion models have this property that you can sort of pick something and then actually change the variable multiple times in the generation process. And so you can imagine for like inverse folding where you're going from structure to sequence, it makes some amino acid, but then it realizes actually that's not really the best thing to fit given some other context. So I'm going to mutate that amino acid back and then mutate it again. So I wouldn't say that this is like an extremely fundamentally always useful advantage, but I think it's an interesting area to explore beyond autoregression.
Okay.
Right, and so this is basically, these guys were very inspired by these papers, and from these papers, they basically said, I think we can unlock guidance for discrete state-space diffusion models. And I'm not going to really talk about flow models. It's easier to think about the diffusion models, but they use the same kind of trick, if you will. And so this has just gone up as a preprint, I think, about a week or two ago. And I'm not going to go, there's a lot, they have a large supplement, a lot of technical details. So I just want to kind of give you the flavor of what's going on, and then you can talk to them or you can read the paper. But basically, these continuous time Markov chain models, they enable diffusion, if you will, for a particular reason that has to do with the tractability of a normalizing constant. And for a similar analogous reason that was not sort of observed by these other folks is it can also unlock our ability to do guidance. And so, if you may have seen the short talk, and I took a few screenshots from this talk. I don't want to go into details again. I just want to give some high-level overview. But fundamentally, in these models, instead of learning the score, you're learning effectively a rate matrix, and the rate matrix is just related to the transition state probability of, meaning that as I'm evolving this process, I maybe change an A to a T or things like this. And so this is the state change, and it's related to a rate matrix, which is like the instantaneous probability in this continuous time. And so in these formalisms that are, again, based, these are the two papers on which this is heavily based by Campbell et al., like the object kind of to think about now is this rate matrix. And these guys have done a brilliant job in kind of pushing through how to do guidance with respect to these rate matrices. And so maybe just to give the one little bit of intuition is that when you're, at the end of the day, you need to make state transitions, and you're gonna, again, you do want to use Bayes' rule. And the main problem in applying that is that you basically need to consider in this version of Bayes' rule, transitioning from imagine I have a sequence of whatever length k, and now I ask how many other sequences could that turn into? If I don't constrain the transitions in any way, then it's like combinatorially large, and therefore there's combinatorially many terms in this normalizing constant, and this breaks everything. And I don't get the fix I get from usual score-based models because I can't actually use the score-based version of Bayes' rule. I have to actually use Bayes' rule. And so that's the kind of fundamental trick is the fact that these continuous time Markov chain processes, they have this assumption that in infinitesimal time, you can only make a transition in one of your coordinates, if you will. And so that may sound like a dramatic constraint, but it's in infinitesimal time. And as soon as I go to any finite time, I can have all that combinatorial explosion again. And so I kind of get in a sense, I get the full capacity of the combinatorial explosion, but by thinking in infinitesimal time steps, I get some computational tractability. And so I think that's as much as I'm going to try to say about that. But what I will do is just show you the two kind of outcomes of churning through the derivations for this. So in the standard score-based models on continuous spaces, you get this sort of unconditional score, and then you add some amount of the conditional guidance score. And here what you get is the log rate matrix is related to the unconditional log rate matrix. And then what you get here is you get essentially a log ratio between how good your current state is at the conditioning variable that you desire versus another x that's infinite, like has evolved for infinitesimal time. And you end up with the log ratio of the density if you have this predictor in hand. So this is predictor, or what I guess we changed it from classifier guidance because we're also using regression models and so forth. So we just call this predictor guidance. And so this is the analog that comes out. And there's also, if you're familiar with the sort of classifier-free guidance, there is a similar analog where in the regular scenario, you're used to seeing the unconditional score getting blended with the conditional score that you seek. Yeah. Yeah. So instead of learning a score, you're essentially learning a rate matrix. That's only in the sense of it being a rate matrix, which has like constraints built in for like conservation of flow, but that's just sort of the definition.
There are these two frameworks how you can learn them, right? So both from Andrew Campbell. And in the diffusion case, like the forward process you pick will also have an influence on the rates that you get. But in the flow-based picture one, you can even exchange you can still change things later on. So like it's not heavily constrained, but it will fulfill like the constraints you expect from a rate, as you said, summing to 0. This is baked in. But how you learn it is like Andrew Campbell's work, not ours. Okay. And the predictor, you just learn a noisy example similar to the standard classifier guidance.
But I guess you are learning all of the transition rates and then including the kind of self-transition rate.
Yes.
Through the loss function. So there is this normalization that you have to do to make sure things sum to 0, but all of those rates are going into some loss function.
And it's interesting because the problem of pushing a dynamics towards an expected value of an expectation is a very, very old one in StatMech. And there's like a huge literature about biasing rate matrices and how to deal with that based on large deviation theory. And so
you mean about wanting to bias them or how they become biased?
Well, so it's you want to bias them.
I see,
so
yeah.
The goal of these calculations is to map out the probability distribution of an observable. But what you do is you construct essentially a tilted rate matrix, which realizes it's the dynamics closest to your original rate matrix that realizes some rare value of And so, like, in some sense, that like it's called a Doob transform. It's like statistical conditioning essentially.
It sounds like we should talk more, and especially you should talk to them. But I'd like to also hear about it. But it sounds like you're trying to anchor it on some known one and then kind of update it, or maybe we should leave this for after. Okay.
Yeah, but I think it's like a really, really similar problem. And basically, what you would do is you would construct exactly the term that's like the predictor here.
Oh, I see. Oh, that's cool. Well, we should definitely know about that.
I'll talk about this for a minute in my talk.
Oh, wow. Okay. All right, everybody. Hopefully
Getting into that, like with Tweedy's formula or
this
is like sequential Monte Carlo
Doob conditioning, right?
Yeah.
All right. Thank you. Thanks.
Sorry, getting it from all sides here, right?
No, that's great. I mean, that's like the point of a workshop, right? Anyone in the middle? Okay. So those are sort of the main results. And then they've done some experiments, including on text, just to anchor it on another paper that was there, but also because in principle this can be applied to text. And then there's sort of an example based on using smiles and small molecules and conditioning on some of the things we've heard before, like the number of rings and the lipophilicity. Just as a toy example, we're not taking this too seriously, just to show that it can be done. There's a direct comparison with Stark et al. On designing DNA sequences for enhancers. These are the folks who use diffusion in the multinomial probability space. And then I think the one that's maybe, well, I guess that is a real problem. I actually don't know too much about this problem, but Hunter kind of came up with this idea that inverse folding models go from, you give it a 3D backbone structure and you get a distribution over sequences, but it's not necessarily the case. Like the sequences may really, in the end, if you put them through AlphaFold 3 or build them and make them in the lab, like they don't often necessarily fold. And so he had this idea that maybe we can sort of make them stability aware by using a stability predictor model. And so this is sort of, I think, like kind of the highlight of the experiments, perhaps in terms of like an actual real use case. And I think actually just as like, I think days before this went on the preprint, there's another group, I think Brian He et al. Doing this with DPO. And so you can, and then you can ask again, like what are the pros and cons of both of these approaches and so forth. But, and so I'm not gonna go into the details here, but I think like this is clearly, this machinery works and it may be related to things we didn't know about. And I think it's useful to have in the overall toolbox of trying to do conditioning when you want to plug and play with one unconditional model that you've spent a lot of time on. So, okay, so yeah, I had no idea how long that would take. So I had another section of my talk, but I think that, there's like still some time left. Let me first see if there's questions, and then if there's not a lot, then I'll try to do the last part of my talk a little bit quickly. I just don't want to rush.
Did you actually try DPO when you, like as a comparison?
No. But I think it's interesting, just to think like, well, theoretically, what are the kind of pros and cons, and even might you combine some ideas from the two and so forth?
Admittedly very different tasks, but when we have tried it, essentially the tendency to like maximize the margin between the probabilities tends to break things on our ends.
Yeah, that's very satisfying, because that does, like, this stuff came, I think, like from OpenAI, right, through the human reinforcement learning, where they're just kind of getting human feedback, but we don't have human feedback. We have like quantitative labels. Like, why would we do that?
I would just add, this is another kind of interesting example to think about autoregression, because often in inverse folding, you're not just designing the whole thing and you want to condition on some subset of the sequence. And so for the He et al. Paper to get DPO to work out of the box, they use this ESM inverse folding because it's like the standard inverse folding model that goes left to right decoding and just does autoregression. And I think it's just one example where maybe like you can kind of fix this with MCMC, but it's not always ideal.
Okay, I'm gonna like completely switch gears and tell you about like a real in the lab project that came out with like a thing that people use now to do protein engineering, but like totally different flavor. And so this is a multi-year project with Dave Schaefer's lab and led by postdoc Bonnie, who now has her own lab in Hong Kong, and David, who's now at Dino, and Cosmo is not on the job market. She is at Dino. Did not update this. Sorry, Cosmo. Maybe she wants a new job. Huh? Highest bidder, highest bidder. And so what this project was centered on was Dave's sort of like a pioneer in using directed evolution for AAV, the adeno-associated virus, which probably everybody here has been exposed to. It's a non-pathogenic virus that has shown a lot of promise and more, even for a gene delivery therapy. So you use the virus as sort of like a delivery mechanism. You put what you want in it, and then you want to get it to the right cell and not to the wrong cells by leveraging the machinery of the virus. And so, so the idea is, in this general space of engineering the protein, you're engineering the capsid, the part on the outside, which is what dictates kind of where it goes, what cells it gets into. And there's some cells that it it's going into it shouldn't go into their cells. Right now, a lot of our ongoing work that Bear is leading is about and Stefan, is how do we get it into like certain cells in the brain. So there's been a lot of promising trials, and in fact, Dave actually has a company that has already gone through FDA approval for this for something to do with some blindness in the eye, and all the successes so far are actually in the brain because of the blood-brain barrier, and the antibodies don't shut it down. Right now, if you were to inject these AAV into somewhere where outside the brain where the antibodies can come, it will just shut it down. And so actually that's one of the engineering challenges, is how do I re-engineer the capsid so it doesn't get attacked immediately before it can do what we want, right? And so that's for that reason, for example, and there's many reasons, but many disease targets just can't at the moment. And I should say, I forgot, Sam's gonna talk later, like this is what his whole company does. So actually he can also tell me if I say something wrong here. But right, so you may have inefficient delivery to the cells, or they might be going to the wrong cells. You might have some pre-existing immunogenicity and things like this. So the first project we had, this started like right when I arrived. This is a very long project. And so basically Dave and I met, and he said, you know, when we start our directed evolution campaigns, we have a few different ways. So for those of you who don't know directed evolution, you basically start with, like, let's say one AAV sequence, and then you induce variation in it to sort of simulate evolution. Then you measure among all those variants, the ones that are kind of closer to what you want, you keep those, and then you mutate them again. So it's like you're creating your own evolutionary path over rounds of libraries of proteins that you measure in the lab. And so they have some starting libraries that work really well for them. Like, so you cannot vary, you can't really vary the whole protein because it's sort of so long that you get such sparse coverage you couldn't learn much. So typically in modern day directed evolution, you're working on some window of the protein, maybe it's 30 amino acids long or something. In this case, they had already identified a 7 amino acid long window that provided really good phenotypic variation. And so this particular project was focused on those 21 nucleotides covering 7 amino acids. And so what they would do when they started the campaign is like kind of, it's almost like the beginning of a active learning scenario. Is they just say, well, I don't know what's going to be best for the thing I'm seeking, so let's make it uniform. They don't make it quite uniform. There's a tweak to reduce the probability of stop codons, but for all intents and purposes, you can think of it as kind of uniform, which is this NNK library. If it was truly uniform, the NNN, those are symbols for different kinds of wildcards. And so the game here, and this is also like, what I'm telling you here is a very simple version of a problem we've thought a lot about in a lot more complexity than is revealed here, mostly because that work's not out yet. But the idea is that how do I design whole libraries of sequences? One way to think about it is I could just give you a list of all the sequences you should go print in the library. If you can measure 1,000 things, then here's my Excel list. But actually, in the modern era, it's one, expensive to synthesize full-length genes, or it's complicated potentially to insert things in. And two, sometimes it's just much, much easier to have a stochastic mechanism in the lab rather than to synthesize them. And so a lot of the computational thinking here is trading off actually like a cost-benefit analysis of like how we're going to actually generate a library in the lab. Is it going to be by specifying the exact sequence, or is it, at the end of the day, what it actually is as a machine learning person is we're designing distributions over sequences where the parameters of the distribution are dictated by the stochastic mechanism in the lab. And this is a simple example where the probability distribution is the marginal probability at each position of each nucleotide. And so, so they started with this uniform one, but the problem was like, the way to think about a directed evolution campaign is like, this is a bunch of lottery tickets, and you want to have the best set of lottery tickets, but you don't know ahead of time what you're designing for. So what can you do that's better than uniform? Well, it turns out that when you use the uniform distribution, you immediately lose half your lottery tickets because those AAV capsids don't function. They're just sort of dead from the get-go. And so this game here was like, can we use machine learning and their data to design a new starting library that's good for all downstream campaigns that want to use that style of library, an insertion library, such that we get the most diverse lottery tickets possible, subject to the fact that they actually package and are viable. And like, I'm out of time, so I'll just say over years we did a bunch of stuff. We tested in the lab, the models, And like the kind of, and maybe I'll just highlight two things. One is that there's a number of people who do design and they use some kind of procedure that they don't understand, and then they post hoc say, well, what was the diversity of the library I designed? And this is crazy, right? Like you want to be able to control the diversity and think about it in a rational way. And so it turns out you can draw a sort of Pareto optimal frontier where you're kind of changing the diversity, and this can be helpful. To guide you to what kind of library you want. But the sort of punchline here was that after we designed this library, we then used it for a selection task that the whole procedure had been unaware of, because that was the point, was to get the best lottery tickets for kind of any arbitrary downstream task. And so in this case, it was to infect brain cells. And this was the standard library at the time. This is a bit of a complicated axis I won't go into. But basically, you can think of this as how efficiently did this starting library infect those brain cells versus our machine learning library that was totally unaware of these brain cells and had zero information, but is just doing better because it had a better set of lottery tickets that it didn't waste. Okay, so I'm totally out of time. And so that, yeah, and then I had forgot to take out those other things. I'll just skip to like the last slide. And then if there's one last question, then we can go into break. But I think these are like very exciting times. I didn't rant about it here, but I've ranted about it a little bit in a commentary of like, you may notice nobody here claimed they were using ChatGPT to solve science, and I think that's a good thing. But there are, you know, whispers out there that we can just use either ChatGPT as it is to design new things, or we just need to kind of train it, and then we're going to unlock the same kind of level of, you know, poetry and insights that we get from ChatGPT. And I think fundamentally, I have like a kind of, also is we just have far less data in almost all of the sciences, and it's I don't have it. I have a chart somewhere with the numbers. It's pretty interesting to like look at how much data, you know, like they ran out of World Wide Web data, and when they ran out of data, they were already like, you know, 6 or 7 orders of magnitude more than what we typically have in any of the sciences, and yet they're freaking out because they ran out of data, and they're in this, you know, kind of clean structured text domain. So I think this idea of predicting the function or connecting sequence and structure to function that's not mediated primarily by structure is still like a really hard one. Like, and I guess catalysis is the kind of the main example. And then as someone in the area, like it's just like whiplash. Like there's so many papers. Some of them are great and inspiring. A lot of them are terrible. It's so hard to keep up. It's so hard to know, like, if any, like, was any of these comparisons, like, actually, should I trust them? Like, you know, like, it's so hard to know what's going on, but it's fun times. And I'll stop there. Thanks. And we'll reconvene at 10:30 for John's talk. Not that John, other John.